Assalamualaikum and a very good afternoon, ladies and gentlemen. Just now was the video from our sponsor of Virtual Science Fair 2021. We would like to express our sincere gratitude to Kenanga Investors Berhad for sponsoring us throughout the event. Ladies and gentlemen, welcome back. We meet again in VSF Science Exhibition brought to you by Virtual Science Fair 2021. My name is Amira Ayanti and I will be the MC for today. It is my great pleasure to welcome all of you to the VSF Science Exhibition 2021, and I think we have now refreshed and ready for our third student bodies of the day, microbiology. For your information, microbiology is the study of microscopic organisms that includes disciplines such as fundamental research on the biochemistry, evolution of microorganisms, and many more. I believe the audience are very curious to know further about this. Hence, without further ado, I would like to welcome Mr. Wan Amirul Shazwan bin Wan Ahmad Munawar and Mr. Muhammad Ajifai bin Muhammad to conduct their sharing session.
Hello everybody and welcome to the Science Fair conducted by the Virtual Science Fair community. Thank you for the MC for the introduction and also inviting us, me and my friend Wan Amirul, to be a representative for the microbiology course. Okay, so a little bit of info. For you all. Microbiology and molecular genetics are actually two separate independent entities. So before this, we have a course called microbiology and also molecular genetics. But starting from 2021 and onwards after this, the university decided to fuse both of these courses and to merge into one singular course, which is now named as microbiology and molecular genetics. So what is actually microbiology? Okay, so microbiology coming from the word micro, meaning small, and also biology or study of organisms. So combining these two words, we come to the meaning of study of small organisms. So what is actually small organism? Is it the bugs or the ants that you all see, throughout your walks beside the forest? Microorganism is actually very, very, very small organisms, and this is a live organism. Okay, so it's a very small that we cannot see with our naked eye. We cannot see using glasses or just watch on the table and then you say, oh, there's a bacteria, there's a microorganism there. No, so microorganisms is very small that you have to use a specific tools in order to identify or in order to properly see them. So I'm sure that all of you are familiar, at least in the science stream students, familiar with the microscopes. So microscopes is the what we call as the propeller to how we actually know about this what we call as microorganisms are there. So microscope, it actually enhances your what we call as sight ability in order to see these creatures. So when you study in microbiology, you will delve more into this word of small. So this word of small is not only like a specific environment. No, it encompasses all, considering human health, animals, plants, even food that you are eating, at the sea, in the land, on the land, terrestrial, and also the technology that revolves around it. So microbiology will be playing around all this type of environment in order to assess their availability on their habitats or their function on these settings. And when we talk about microbiology, it is most obviously a research based study. So because how you see the technology is always evolving, like the humankind always like coming up with something new, coming up with something novel, so does the microorganisms. So microorganisms is following us, like we evolving into a very like we adapting to this world right now. So these microbes are also adapting to this all of the factors and patterns that important for their survivals. So it is important to us, for those who are studying microbiology, to keep up to date with researchers. And you can see, like in this current time where the pandemic of COVID-19 is rampaging. So as a microbiologist, you must know what the situation is, how and why all of this happens. So all of it are included in the study of microbiology. So as you can see in this picture is a picture of a researcher with an agar plate. So those little patches of fluffy white things is what we call as microorganisms. So those are actually a fungi colony, or colony is a group of these microorganisms. So you think you saw if you saw a rotten food or rotten fruit, you will saw these patches. So actually, as a microbiologist, it is very in our interest to actually study them, and also to attempt to grow them and maybe manipulate them for our benefits. So like some of these microorganisms may produce something that is good for us, like the antibiotics. The antibiotics that you take to treat your infections are actually produced by these fungi. And there's also importance of diseases, and playing with other what we call as metabolites to use in other industry. So to summarize all of this in short, microbiology and also genetics are actually, we can say that it is like playing with small things that are invisible to the eyes. It's a very small, it's very precise, it's very complex. And obviously for the genetics, for those who are studying DNA material, RNA, they unlock these secrets of what is the meaning behind all of these codes that are transcribed onto each organisms. So all of these living organism, including us, because we are actually composed of a little cells, are possess this genetic material. So for those who are interested in genetics, like to manipulate them, are very revolving around this. So if you are a keen game player, you are a science fiction fans, see movie and novel, you will always see that in example of like a zombie apocalypse. Okay, so most of the time the causes will indeed be these microorganisms. Why? Because they can actually alter the what we do, and alter what the our action and our whole survivability. So like, of course, the logic explanation, if a zombie apocalypse will occur, it must be caused by these harmful microorganisms. Or there's also a theory like this, where there will always be a theories of a what we call as candle theories, where if a pandemic occurs, it must be someone's someone's fault. They alter those genetic and they make it as a weapon. Who knows? But this is actually can happen. All of this in the genetic picture. So next, I will go to a facility provider, and I will pass down to my friends, Juan Amirul, to explain more about what facilities are provided in our buildings.
Okay, thank you, Faiz. So now I will show you the facilities provided by in the microbiology and genetics building at University of Malaya. Okay, so now okay, this is the surrounding of the buildings around. As you can see, the first picture, this is the main building of the microbiology and genetics building, where this building is located in front of the our main hall, Dewan Tunku Chancellor, and near to the University of Malaya field. Okay, so the second picture, as you can see, this is the opposite side of from the first picture, where in the second picture there is a surau at the corner of the building, where it is easier for Muslim students to perform prayer during the break or lunch. Okay, next. Okay, and this is the main lecture hall for students for meetings, lecture, and so on, or seminar. It will be held here, and you can see it's really conducive, and sometimes that you can catch cold in the hall. And next slide. Okay, this one is next to the main lecture hall. Before this, the day one kuliah 16, and the next this one is day one kuliah 17, where most of the lecture or classes will be held here during early morning until usually until afternoon, something like that. And as you can see, this we sit on the sofa, and yeah, sometimes once you get sleepy during the lecture, but yeah, all you need is to be prepared to be in this type of lecture hall, because this is the modern lecture that sometimes make you harder to study. But don't worry, it is conducive and really good. Next slide. Okay, this is the hallway near to the one kuliah. This is the hallway at the mid of the building, center of the building, where here you can buy the drinks from the vending machine, and you can gather here for discussion and so on. And the next picture is the convenience store, where this is specific for the foundation student, but it's located in this building, so you guys can buy stationeries or food from here for lunch and so on. And this convenience store provide you variety of food also, because you can get nasi belau or nasi lemak only for RM1.50 here. And there's also you can buy a lab coat or something related to your studies here. It's provided. Okay, next. And during practical session, this is the lab where you spend your time almost every day, I guess, 3 to 4 times per week in this lab doing your practical session in the afternoon. Okay, and as you can see, in this lab, they are provided, each table is provided with the microscope and Bunsen burner, where this Bunsen burner, you will use it almost every practical, because it is it is compulsory for microbe to maintain cleanliness in the laboratory. Okay, next slide. Okay, this is a M2 laboratory. This lab is located next to the M1 laboratory, where we can see the differences. This M2 laboratory use this type of chair, while the M1 laboratory, see this? Okay, M1 laboratory are using another type of chair, which is just so we can see. Okay, and yeah, this is the place where you guys will do a practical session throughout the degree courses, about 4 to 5 semesters in this lab. Okay, next slide. Okay, so now we proceed with the challenges and how we want to overcome these challenges during the study in this process. Okay, the first one is the maintaining the aseptic technique. So what is the aseptic technique actually? Okay, aseptic technique, you will apply them during the practical session. And aseptic technique is the practices and procedures in order to prevent the contamination from the microbe or pathogens. And it is important in order to minimize the risk of infection. Okay, for example, we need inoculating loop, as you can see in the right hand side upper picture. Okay, this loop is essential for you to take the bacteria from culture and to transfer them into agar. Okay, but aseptic technique that will be used is you need to heat up the loop, and you need to cool down it for some time. Then you will take the colony of the bacteria and transfer it. If you don't do this aseptic technique, the result will be affected. Okay, next slide. Because as you can see in the picture, contamination may occur. So we can see in the first picture, there is a fungi or mold grow in the agar. So this agar is contaminated with the mold. And the second picture, as we can see, this mix of bacteria colony and also fungi colony. Because so, if you want to focusing on getting the bacteria, but if you do not do the aseptic technique properly, this is the result. It will be affected. You will get both fungi and bacteria on the same place. And also, if you don't implement this aseptic technique, risk of infection uh, can occur because sometimes you will deal with pathogen such as bacteria that causes diarrhea, or maybe you deal with virus that can cause disease. Next. Okay, the second challenges is as I said, you will study in a really conducive lecture hall. This is this conducive means really cool lecture hall because so I suggest that you prepare in order to study in this cool lecture hall because you will be in this lecture hall from early of the morning until afternoon, sometimes until evening, almost 4 to 5 days per week. And you must be prepared, bring your sweater or wear your socks because some people cannot handle this type of cold. Okay, that's that. Okay, the third one is during practical, sometimes this lab manual actually doesn't help at all because some lab manual not provide you the detail of the experiment procedure, or maybe it just give you the rough idea but not the detail of the experiment. Okay, so I suggest that you to always communicate with your lecturer or lab assistant because they are friendly actually. Because when you always interact with them, you will find it easier for you to do the experiment. And since you will do the practical 3 to 4 times in a week, up to 4 to 5 semesters, so actually it does really help you in networking and also uh, enhance your comprehension regarding your practical and courses. Okay, next slide. Okay, the fourth one is since we now in the midst of the pandemic, online distance learning is essential because almost all courses or university are implement this type of learning. So you will have these courses, microbiology courses, you will have classes from morning until evening, right? So you need to manage your schedule well because you will be in front of the laptop from the morning until evening. So sometimes you might become really stressed or maybe sleepy, lost of hope, and so on. Therefore, don't forget to spend your leisure time with your friends or family, such as playing games or uh, gather with them lepak-lepak as you always do lah. Okay, next slide. Okay, the fifth one is the miscommunication. This miscommunication will always occur between you with your classmate or between you and your lecturers. And so you should know how to have a proper discussion with them. Arguing or holding grudge towards them is not good at all because you will study and learn with them for the rest of your degree studies up to around 6 to 7 semesters like that. You will always be with them. So discuss properly with them, or if discussion doesn't help at all, don't worry, there will always be a channel or medium for you to suggest improvement or something that related to it. Okay, thanks. Okay, this courses microbiology is not same as another courses such as mathematics or engineering or physics because these subjects will require you to memorize a lot. Okay, and most of the subject in this microbiology and molecular genetics courses involve a lot of memorization instead of calculation. So, because you need to understand those particular processes of bacterial metabolism, biochemistry, and you need to memorizing a lot of terms such as the the microbe or bacteria scientific name, the name of the instrument, the specific molecular substances involved in the process, and so on. Okay, so I suggest that you use mind map or acronym. So I believe that all of the students, since primary school or secondary school, you already learn this type of mind map or acronym. So implement this. Be familiar with this kind of studies will help you to memorize a lot of things, and you should know that because the lecture notes or the references provided for you to study sometimes are too complicated and too detailed that you need to something that can be comprehend or digest easily, such as this type of method, such as doing mind map or acronym. Okay. So now we proceed with job opportunities, which will explain by Farid. Thank you.
Okay, thank you, Wan. Okay, so I will proceed with job opportunities of of what these microbiologists or also geneticists will be working on if you will be you will graduate, and what will you be working on this working environment. So when we think about microorganisms, of course it is like almost indefinitely separated from all four of these major field. So the first one being clinical medical health, and so you can work as a hospital microbiologist, or if you are aiming to be an expert, you might be a immunologist or parasitologist, virologist, mycologist, and others. So because of this microorganism actually have a very big impact on our health, okay, like TB, tuberculosis, that is caused by bacterial infection. This COVID-19 is actually originated from the virus of coronavirus 19. So those who are, those doctor who take your samples and to check, they will send to the lab, and because the doctors are the one who are what we call as analyzing the symptoms and such on, and will be prescribing medication later on. But the one who are analyzing what is the cause, or the causes of these particular infections, be it this species or that species, is the microbiologist. So those samples will be sent to the lab, and these microbiologists will be conducting several tests and other mapping or like sequencing to know who actually involved in this infection. So if you are interested to working in a hospital environment, maybe in a medical environment, but you did not want to be a doctor, maybe this is a calling for you. And when we talk about medicines, there is and there will be a synthesizer. Okay, so those who are coming up with this new medicines or this new medication source will be the pharmacists. Okay, and usually pharmacy will be working hand to hand with these microbiologists, because you will need to find sources of where this raw material are coming from, and most of the time are the microorganism itself. So take antibiotics for example, like the earlier I give you. So antibiotics is produced by fungi. Okay, there's several types of fungi that will be producing these antibiotics, and why they are producing these antibiotics is because they want to fight with this bacteria in order to these are their territory. So they produce these antibiotics to fight those bacteria if we are talking in normal settings, and then we take that advantage. Also, we know that this fungi will produce these antibiotics, so maybe we can take them and we multiply them, we grow them, and those products we will be extracting and will be used for our benefits. So the search for new medication, new antibiotics is going on and going on, and need to see towards new, to find new species in order to see new resources. So that is for clinical, medical, and health, environment settings. So the next one will be industrial. So if you think that microbiology is only related to this, medical things, it's actually quite wrong, because microorganisms are everywhere. Microorganisms are affecting everything. Even in industrial itself is a very major point for microorganism to be involved in, and that's where our expertise will be needed. Okay, so take quality control as example. Usually in food factories, in pharmaceutical companies will have a quality control segment, and usually there will be a microbiologist or some microbiologists that will be observing these products, whether we will take food as example. So whether this food is safe for consumption after packing, is the count of is the bacteria or microorganism inside the food is safe for consumption, is there any leakage, is there any contamination happens in because if there is a contamination, then those products that contain this microorganism will be consumed by the consumer, and you will get, food poisoning, and of course there will be thousands of dollars suits will be getting into you. So you don't want that sort of trouble. So this is why quality control is important. So there's also a biotechnologies, microbiologists and geneticists can also be a biotechnologist. Okay, so you can alter these microorganisms maybe to produce something, to produce acids, to produce enzymes in order to use in industry, like in paper industry or in clothing industry. So you can actually search this in the web, and you will see, oh, this is a very diverse thing actually. Okay, so the other example that I want to give is the fermentation scientist. So fermentation is a very big aspect in most of the economy. If we're talking, like of course Malaysia is a Muslim country, but like the wine industry of the the West, or the beer industry, the alcohol industry, will be relying on this fermentation scientists, because you want for the fermentation process to be precise, to be in low cost, but in a short time. So how do you want to achieve that? You must consult with these scientists. Okay. And of course, because of microorganism is actually, microbiology is a subject that is revolving around theories, around facts, established data. So being a researcher are most of what most of the microbiologists will want to be. So they will be maybe working in the university as a lecturer, as a professor, or maybe as a lab assistant, as a as a paid researchers, and inside research and development, like in the government sector, or in like MARDI, the animal sector. So you have these microbiologists to conduct research, like. Take example as like for the farmers, like, oh, there will be a what we call as pandemic, but a outbreak toward the paddy plants. Okay, so if this happen, then the economy of our country will plunge down. The food source will be endangered because of how much the loss will be. So you need all this expertise in order to avoid these things to happen, to treat them, and also to improve them. How to making a better crops, how to make a more resistant to weather, or how to make them produce more abundant produce. And maybe it's in a soil where we will learn about how to remediate the soil, like, oh, this soil is so toxic, so how you want to treat them. If there's a cases of people people illegally dump all of those toxic material toward lake or sort and so on. So you have this need for research to how to overcome or how to properly eliminate all of these factors. And the other one is field professionals. So field professionals is professionals on each field. Okay, so maybe if you are working in the government, you may be an epidemiologist, or maybe you want to be a cosmetic technologist. You are a professional with these cosmetic microbes. For your information, like our skin is actually a barrier from the outside of the world. Okay, so like the community of this microorganism is actually being affected toward our health and our skin. So nowadays, you might know about the cosmetics, especially girls, about the prebiotic based cosmetic or probiotic based cosmetics. So those who are come up with this formulation is actually the cosmetic technologists. So you might be, for those who are in what we call as government sectors, like those are professionals where they, especially epidemiologists, where they analyze why this outbreak happened in Malaysia, how to curb all of these, the steps to avoid all of this coming from them. Okay. So that's all for the job opportunities. Now we will be moving on to the question segment. So the MC. Okay, so how difficult is it to practice aseptic technique for a first timer, especially after having an online class session? So I will give one to answer that.
Okay, aseptic technique for first time. Yeah, usually during our last session, for the early semester, yeah, it is sometimes difficult, but you don't know what is the best timing for you to let your loop to be cooled down, when to take your microbes, when to culture them, or some aseptic technique. Sometimes you forgot to disinfect, to clean up your bench during the practical session. So for the first time, yeah, usually we forgot, and our result will be contaminated with mold, with fungi, and we didn't get result as we expected. But throughout the semester, we will learn, because, like I said, we have 3 to 4 times practical per week. So in the third or fourth week, you will be used or familiar to those techniques, and somehow you will get the best method, and it will you can get the result as you expected. But during the online class session, you will teach by video, so it is not similar as hands-on, unless you practice that at home. So yeah, you need to. This is the disadvantage of the online class session, because you don't have the hands-on practices, and then watching videos might be somehow help you in the future lab or practical if the pandemic is over, inshallah. Thanks.
Okay, so the next question, did you guys deal with virus/virulence pathogens in lab, or strictly to prokaryotes / simple eukaryotes, yeast or protists? Okay, so these labs, we have what we call as BS, or biosafety level of labs. So in our buildings, we have the max is second level BS2. So BS2 is like there is a very specific microorganism that will be allowed to handle. So for like dangerous pathogens, like for those who are in should be handled in BS3 or BS4, will not be handled in the BS1 or BS2. So we don't do all those very dangerous pathogens. We only play with very mild, you know, very mild microorganisms that may or may not cause infection, if only you are practicing aseptic techniques. So most of the time we are playing with yes, we play with prokaryotes, we play with simple eukaryotes, we play with bacteria such as Escherichia coli, we play with sometimes we play with what we call Streptococcus and all of that. But you need to always be aware or always be in a safe environment, because although you might think, oh, this is just a mild pathogen, but it is actually a can be a bigger problems when it enters into your body, and you will, there's accident happen and your body are not functioning properly, and so yeah, you might end up in a hospital. So that's you must always ensure for safety and aseptic technique to be well practiced all of the time. Okay. Next question. What is microbiology specialties compared other biocourses like biochemistry or ecology, etc.? Okay. In Biology Institute of University Malaya, we have several courses that will tackle very specific field of study. So for biochemistry, they will not delve on how this microbe maybe what we call as reproduce or enter the body. Biochemistry will focus on they will focus on how the process of the metabolites producing, how they produce enzymes, how they produce acids, and so on. But there will be a little bit of what we call as connections between each other. And for microbiology, there will still be biochemistry aspect of the biochemistry, but we will not delve too deeply into the settings. There will be a very specific setting for us, like for the basics and for the how this bacteria interacts, what will happen if this metabolites is produced, but not necessarily we know how this reaction occurs very deeply. Same goes to ecology. So ecology may learn about fungi, may learn about bacteria, because those microorganisms also affect the ecology of the world. So although they not delve too deeply into ecology, but there's still a connection between microbiology and ecology. Same goes to those who are studying microbiology, although they will not delve more into those type of ecology, like interaction between this animal and that animal, but there's a interconnection also with ecology. So like if this animal interact with this animal, does the microbes changes or and so on. Okay, so the next question, I believe it is the last question. Okay, so what is difference between microbiology course and genetics course? Okay, so before this, before 2021, microbiology and genetic course are two different entities. So the microbiology will learn more on aspect of microbiology, those are microorganisms, and sometimes, only sometimes, we will have a little bit of connection with genetics. So we learn about genetic material, about if this microorganism evolve, how they evolve, how they take the genetic material. So these are what those genetics peeps are learning, and the genetic peeps are also learning a little bit of interconnection with microbiology. So because we are learning about this DNA, so how do we manipulate this DNA of that certain microorganism? So that is the difference. One delve into like this sort, and the other are this sort that we have a little bit of interconnection between each other. However, for after 2021, because the courses are fused, so that means you will be learning into both of this field, and so will be increasing your your knowledge and also your availability in the market later on. So I believe that's all for the question segment. So moving on, we will be wait a second. We will be going into insights we explore segments. Okay, so first of all, I will explaining my part of what I want to share with all of you, and the next one will be my friend Amirul. So, are you guys ready? I hope so because I will be sharing this. Okay, so I believe that all of you, if not all of you, have drink at least one of this type of cultured milk drink. Okay, so at the left we have Yakult, in the middle we have Betagen, and oh, I'm sorry, I supposed to put the right one is to be Vitagen, but I'm sorry for that mistakes. But the similarity between all of these three drinks are actually you are drinking a milk that contain microorganisms. So if you did not know about this beforehand, like, oh, I think I just thought that this is a normal drink, the flavored milk drink, but no, it's actually containing billions of microorganisms that you are now ingesting and putting in your body. So interestingly, these microorganisms are actually beneficial for your health, and this is what they claim. This is what the drink companies claim, that drinking this will be good for your guts, will be good for your the dialogue, like perut sihat, badan sihat, or something like that. So these bacteria are what we call as prebiotics. Probiotics, I'm sorry. So probiotics is a beneficial microorganisms that will induce a very good effect on the human host or the host. So you see here, this is the ad for Vitagen. No, I'm not sponsored by Vitagen, but I'm only borrowing the ads from them. Okay, so they say that Vitagen boosts your immunity with probiotics. So how actually ingesting microorganism improving your immunity? So this is actually a very interesting research inside the microbiology. So our bodies, in our guts, there's thousands of types of microorganisms that interact with each other. So these beneficial microorganisms actually helps you to regulate all of these immunity products. So they will produce products that will help our body. They will also suppress all those harmful microorganisms that is in the gut. So by ingesting all of this, you will increasing more on the good side rather than the bad side. So the good side then will fight with the bad side, so conquering a very good health for you. Okay, so next we'll pass to my friend Wan Amirul to explain about his sharing.
Okay, thank you, Faik. So now, as we know, microbes or pathogens also can cause serious disease or infections toward us. And for example, in this picture is the chickenpox. Okay, this is chickenpox. I believe most of us have been infected with this type of virus during our childhood, right? So this chickenpox is a highly contagious disease which caused by a virus called varicella zoster virus, VSV. Okay, and the disease result in a skin disease infect skin, and the skin that form rash or small or itchy blisters on our skin, the reddish one that we can see on those pictures of the cute babies. And as this chickenpox is an airborne disease which can be spread through aerosol, and it can be spread from one person to another person through the cough or sneeze from an infected person. And this is important to know about this type of pathogen, the virus itself, because we will know how this varicella zoster, the mechanism, how they infect the host, how they can be treated, or how they can be prevented, and we can implement the healthier lifestyle in our family or our community in order to avoid this type of diseases. So what we should do is we need to always practice cleanliness, and don't worry, right now we have the varicella vaccine in order to prevent this type of disease. Okay, another type of disease that can caused by the microbe or pathogen is scabies. Scabies is a skin infection which caused by a mite known as a Sarcoptes scabiei. At the bottom of the slide, you can look at the name, Sarcoptes scabiei. It is a specific name for the mite. And what this might do, they reproduce on the surface of your skin, and then they burrow it and they bury their eggs into your skin. And this causes an itchy reddish to form on the skin. And these bugs are so small that you can't see them with your naked eyes, but you can certainly see their effect when they reproduce on your skin. And it can be spread by skin-to-skin contact, and also can be spread through your clothing or your beddings. Usually, it is popular among the students from boarding school, right? And in order to prevent getting infected by the scabies, yeah, as we know, it is a skin-to-skin contact. So we need to avoid contact with those infected person, and we need to avoid unwashed clothes, or maybe your bedding, and you need to always wash them in hot water so that those might can be eliminated. So I believe with knowledge about those microbes or pathogens, on how they can be spread and how they can cause the disease, we will come into a solution on how to prevent those disease and implement a healthier lifestyle in our community. Thank you.
Okay, so we will proceed with quiz time. So to summarize all of these and to make you all lose all of this tension, maybe. So we will proceeding with our quiz. So I hope you guys are prepared. So let's go on to the first quiz. Okay, so the question is: microbiology is the study of A, plants, B, animals, C, microscope, or D, microorganisms. So I will give you guys 5 seconds to maybe type in the answer or think the answer, and we will see the answer later on. Okay. Okay, so I believe you all have the answers prepared if you are, hearing this sharing. So the answer will be the D. So microbiology is the study of microorganisms, which include virus, bacteria, protozoa, fungi, and others. Of course, there will be a connection with plants and animals, but we are not actually studying them. We are studying what the interaction of these microorganisms with them. Okay, so the next question. The facilities below are available in the microbiology and genetic buildings except A, M1 lab, B, Kedai Serpeneka, C, cafe, or D, Dewan Kuliah 17. So same, 5 seconds. So I hope you prepare the answers. Okay. All right, I saw some very good, answers there. The answer will be C, cafe. So although our building doesn't have a cafe, but you can access this cafe in the building near to our building. So it's just a 5 seconds, 10 seconds walk to go to grab your lunch and then resume your study at the lecture hall. Okay, so the next quiz will be quiz 3. Okay, so the question: what should you do to avoid contamination during the experiments? A, hold your breath. B, practice aseptic technique. C, practice breathing technique. D, wear lab coat. Okay, so we will see the answer is B. Practice aseptic technique. So aseptic technique is always the practice of and procedures to avoid this contamination and risk of infection toward us. So you will learn this aseptic technique first hand or first thing before you actually doing any other experiments, how to burn the loops to take the bacteria, how to disinfect your bench using alcohol, how to clean your equipments and hands with soaps and with antimicrobials and so on. So aseptic technique is very important to protect us and protect the microorganism from going everywhere. Okay. So the next question, below are the job scope available for microbiologists and geneticists, except A, doctor, B, quality control, C, researcher, D, lecturer. Okay, I saw some answers there. So the answer for this question is A, doctor. So also microbiology may intrude some with inside the like doctor-like environment, but if you want to be a doctor, you need to go to medical course to be a licensed doctor. Okay, so us as microbiology, we have our specific field, we have our specific jobs, and we actually work with this doctor if you work in a hospital environment. So maybe if your friends are working as a doctor, maybe you can work with them and you will be the lab microbiologist. Okay, so question 5, the cultured milk such as Vitagen and Yakult claim to be very beneficial for our gut health. What is the basis of that claim? Okay, A, it provides vitamin. B, it contains leaf beneficial bacteria. C, it contains antibiotics. Or D, it contains proteins. Okay, so we'll go through the answer. The answer is B, it contains leaf beneficial bacteria. So most of the time, this cultured milk, the main selling point is the live bacteria itself. So if you go to the Yakult, ad in the TV, they will say, this drink contains lactobacillus protectus, or also known as lactobacillus acidophilus. So these are lactic acid bacteria that helps to promote gut health, which can lead to better overall health. These drinks may, included with some vitamins or minerals or some other, good thing for your health, but it is not the main selling point, and it's not the what the company intend to do with this drink. It's just an add-on. Okay. So this is the last question. So question number 6. What is the main cause of chickenpox infection? A, virus, varicella zoster. B, bacteria, Lactobacillus casei. C, virus, variola virus. Or D, parasite, Sarcoptes scabiei. Okay, so I believe all of you are very, paying attention to the sharing given by one. So you must know this answer, and I believe most of you answered the correct, pick the correct answer. So the answer for the chickenpox infection is A, virus, varicella zoster. So for the others answer, Lactobacillus casei is the bacteria that contained in the cultured drink that is a beneficial bacteria. Variola virus in answer C is the main culprit for smallpox. So there is a difference between chickenpox and smallpox. And the last one is Sarcoptes scabiei is the main cause of scabies infection. So I want to just address one of you guys' question, but PhD, of course, philosophical doctor. So you can be a philosophical doctor, but you need to, of course, study hard, study more, okay, and go to a PhD course, PhD path, okay. Microbiology cannot be medical doctor, okay, and not a philosophical doctor. If you want to go to the microbiology and you are very interested in research, you're very interested in all of this, small environment, small animals, small virus, bacteria, I hope you will be a very distinguished professor that will achieve your PhD in a very short time. Okay, so I believe that is all from us and from my friend, from Microbiology UM. I will pass down to the MC. Thank you guys for your time and your keen attention. we see if you want to go to maybe we'll see later on. Okay, thank you so much, Nancy.
Thank you so much. That was such a fun and engaging session. So before we end this, 5801, do you have anything to add? Any
For me,
last
I don't
word?
have.
One?
Same for me. Nothing to add.
Okay, thank you so much. Again, thank you so much to Mr. Wan Amir Shazwan bin Wan Ahmad Munawar and Mr. Muhammad Azri Faik bin Muhammad for the detailed explanation conveyed during the sharing session. Ladies and gentlemen, as you can see in the live chat, there is a feedback form posted by VSF team. Kindly fill up the form as your feedback will be counted in choosing the winner for the best presenter. Before we end our last session for today, let's give our brightest smile for photography session.
Everyone ready? Okay. Okay, give your brightest smile in 3, 2, 1. Okay, wait a moment. Okay, another one in 3, 2, 1. All right, thank you.
Thank you. All right, ladies and gentlemen, now we are done with day one of VSF Science Exhibition. I thank all the presenters and also all of you for attending our VSF Exhibition 2021. We truly appreciate it. On behalf of the organizer, I would like to apologize if we have done any mistakes throughout the event. I hope all of you are able to comprehend many new knowledge and understand more about biology courses in University of Malaya. Don't forget to keep in tune for our next VSF Science Exhibition on tomorrow, 11th, 17th, and 18th April 2021. With that, till we meet again, I am Amira Ariyanti. Assalamualaikum, thank you, and good day. Bye!